Okay, you're all very welcome to this webinar today, to this BioPhorum webinar entitled CGT Method Validation Controls, Current Practices and Considerations. My name's Kathleen O'Hagan. I'm a global change facilitator. I'm just seeing a question. Yes, the slides will be shared on the BioPhorum website, and the recording of the webinar will also be shared. Just I saw that question come in, so yes, that's no problem. So my name's Kathleen O'Hagan. I'm a global change facilitator within BioPhorum, and I'll be hosting the webinar today. But we do have, I won't be the one delivering the detail. We do have some experts in the field. You'll see them on the screen there that will talk you through a recent publication that our team has produced. Okay, so in terms of the agenda for today's call, really the webinar will be split into two halves. The first half of the webinar will consist of an overview of a recent publication that one of our teams within the Cell and Gene Therapy Forum have produced. And this publication is a slide deck that was with the aim to sort of address challenges associated with identifying the appropriate analytical controls for cell and gene therapy products. I believe my colleague has just dropped that link into the chat of that deliverable. So if you haven't looked at it yet, we'd encourage you maybe to download and have a look. But we will be showcasing some of the slides from that deliverable on the webinar today. So following that overview, we'll then move on to a panel discussion where some of the key authors of our published slide deck will be talking through some of the key questions and challenges associated with this topic. And we will, of course, be encouraging questions. You as audience members will be very welcome to put questions to the panelists. We will be taking the questions after the overview and sort of through the panel discussion. So we ask that you drop the questions into the chat as they rise and come into your head, and we will pick them up then at the end of the presentation. Okay, before we do go into the our presentation, the panelists did have some questions for you guys, so the audience members, just to understand the demographics of the audience, and maybe we can tweak the presentation just accordingly. So I'm going to launch the first question. So the first question is just around the experience of your experience in the cell and gene therapy field. So have you been working within cell and gene therapy for 0 to 1 years, 1 to 3, 3 to 5, greater than 5, NA or other? And that question should hopefully appear in your screen shortly. So please answer that and we'll be able to look at that now in a few minutes. So hopefully onto your screen there. And then the next question is, what department are you currently working in? So are you QC, QA, regulatory, analytical development, manufacturing, NA or other? And again, this will appear on your screen. And if you go into the chat, then you'll be able to see sort of the makeup. So let's see what we have still going on. So in terms of experience, so the majority seem to be greater than 5 years. Oh, no, sorry, 3 to 5 years is coming up at 39%. So we've got 13% 0 to 1, 18% 1 to 3, 38, it's going up to 40% 3 to 5, and then 26 greater than 5 years. So thanks everyone for answering that question. And then in terms of department, so analytical, I suppose not surprisingly, is coming out strongest at 32%, and then we've got QC at 26%. So that's great. That gives us an idea of the members of the audience. So thanks for answering that. Very quickly, before we do go into the crux of the presentation, I appreciate a lot of people here are probably familiar with BioPhorum, but for those audience members that aren't, I just want to give a very, very quick overview on who we are and what we do. So BioPhorum's a global collaboration who bring together experts in the field. We're going to come together to collaborate, share challenges and problems with the idea of addressing those problems by developing best practices and procedures as a group and accelerating progress. The Cell and Gene Therapy Forum is one of 11 forums within BioPhorum that focus on specific topics. So you'll see here from the wheel on the right of the screens, we've got development group, drug substance, fill finish, etc. And how the Cell and Gene Therapy Forum differs from other forums within BioPhorum is really all other forums are focusing in on general biologics. So the Cell and Gene Therapy Forum really is of end to end, from development straight through to commercialization, but within the cell and gene therapy space. And this forum, the Cell and Gene Therapy Forum, was formed in 2018. We've 38 member companies at the minute, with over 850 active participants who, and some of these you'll hear from today. In terms of teams then, and how the Cell and Gene Therapy Forum is made up, we've sort of two levels of teams. So we have what we call the high-level work streams, and these are sort of larger teams that meet intermittently, typically sort of every 2 months, to discuss high-level topics and sort of quick-fire questions. And these teams can either be modality-focused or functional focused. And then the second level of teams that we have are what we call our in-depth work streams, or a deep dive. And these teams meet more frequently. They're working on a specific focused topic with the goal to produce a sort of defined deliverable. And it's one of these deep dive teams that you'll be hearing from today. So the team you'll be hearing from today is the Cell and Gene Therapy Method Validation Controls team. And this team really came together to address the question around limited material when it comes to analytical controls, and the challenges in securing that material for your controls. And the second problem really they wanted to address was the insufficient guidance that's currently available. So insufficient guidance around process changes, how much variation is allowed when the process changes, and what materials can be used should the ideal material be limited or scarce. And the goal of the team then was to produce a deliverable that collates all this current guidance and sort of highlights best practices within the industry to suggest a risk-based approach. And that was the outcome of this team, was the shared slide deck that we have shared with you today. So when you download the slide deck, this is the cover slide that you'll see, and you'll see that this work is a collaboration of over 11 organizations, and you're going to hear a little bit from some of the key authors today. So I'm just going to pass over to each of the presenters just to allow them to introduce themselves quickly and say a quick hello. So maybe, Parul, do you want to start?
Sure, thank you, Kathleen. As Estella's Associate Director of QC Operations, my team and I ensure GMP compliant testing for all cell therapy modalities at production in the Massachusetts facility of Astellas located in Westboro.
Hello, everyone. Yep. Hello, everyone. Thank you for being here. I'm Shewit Tekeste, Principal Scientist at J&J, based in Malvern, Pennsylvania. I'll be bringing expertise in early to late phase CMC analytical development, supporting both cell and gene therapy products.
Hey, I'm Andrea Sobjak, the Director of Viral Vector Analytics, also from J&J. And I also have experience across both cell and gene therapy products.
You're on mute, Jan, just to say.
My apology. So good morning, good afternoon, depending on where you are. Thank you for joining today's webinar. So my name is Yan Zhi. I'm the Director of Cell and Gene Therapy Product Owner at CSL, based in King of Prussia. So my current role is to serve as a CMC lead in a commercial organization in order to ensure the commercial supply of Hemogenics to both US and EU market per the approved dossier. In addition, I close work with our regulatory colleagues in order to support the additional submission of Hemogenics to Wave 2 and 3 countries. So thank you for your time today.
And Brian, last but not least.
Hello, everyone. Thank you for joining and welcome. My name is Brian Mosley. I'm a senior scientist at BridgeBio Gene Therapy here in Raleigh, North Carolina. I have the privilege of leading a group. We are the development support team and analytical research and development here at BridgeBio. I've been in the industry for about 25 years now, large and small molecule. I've been in the gene therapy space for about 4 years.
Thanks, Brian. Okay. Just a quick disclaimer before we start. So just to state that all the opinions expressed during this webinar are belong to the individual and not necessarily belong to the individual's employer, organization, or any other individual. And then last slide for me, second last bit possibly. So when you download the slide deck, it's quite a large slide deck. So to help the user navigate through that, we've introduced this navigation hub. So you'll see in the slide deck a similar slide where you can click on the various sections, and this will bring you directly to that slide. And then in every slide in the slide deck, there's this icon, the navigation hub, which if you click on will automatically bring you back into that navigation hub. So the idea is to allow the user sort of to easily freely navigate through the tool that we've produced. Okay, I'm now going to stop talking and pass you over to the first presenter, Parul, who'll talk you through the executive summary.
Thank you all. This executive summary is, as Kathleen said, the summation of a lot of people working together. The presentation reviews the regulatory challenges faced in cell and gene therapy with a focus to identify and use appropriate analytical controls when dealing with both limited regulatory guidance and per the nature of the cell and gene therapy, limited materials. Within the topic of lack of guidance, this slide deck will focus on the following challenges. What is considered appropriate controls and standards? That is, what are the working reference material versus in-house or commercially available materials? What alternative materials can be used when materials are scarce for some of the assays? And if processes change, is the reference standard still relevant? That is, bridging study considerations. All these slides will be spoken in the next few, by the next few people. Thank you.
All right, so when this BioPhorum analytical validation workstream started, it became quickly apparent that we needed to review the regulatory landscape. The novelty of each of our products raises challenging questions from health authorities during filing that relate to the CMC product manufacturing and quality. So we approached this from a developer's perspective. We reviewed the current regulatory landscape related to the release analytics requirements and also the controls. And I need to warn you that the list we focused on was in the US and the EU guidelines, giving the minimum assay and control requirements that needed to be met. The outcome of the study is summarized in this user-friendly slide deck, which based on the poll, it's still going to be beneficial for both the novice and the experts in this field. Next slide, please. So this is just an example of the analytical assay guidance table. You'll find that for each analytic guidance subject, we have introduced hyperlinks. That should take you to the snapshot of the current guidance document. As a caution, these links will not capture any ongoing or future updates to the document, so please keep that in mind when reviewing the slide deck in the future. And then next slide. So the good news, we all love good news, is that at least we have a minimum starting foundation for early phase programs. And as expected, with new modalities, there's always going to be challenging or new areas of improvements. So right now we have areas of improvement that can impact mid to late phase programs. One example on one major area is around the phase appropriate potency requirement. Question arises is, how do you know if your product specific matrix strategy is indeed sufficient for filing? Another area of improvement is knowledge sharing without compromising intellectual property. So how can this be done? This is very tricky. But a couple of years from now, we've seen some roundtable discussions, open forums, facilitated by nonprofit organizations such as BioPhorum and also Alliance of Regenerative Medicine. A white paper just published a couple weeks ago by Alliance of Regenerative Medicine, which summarizes takeaways from a scientific workshop on best practices for analytical methodologies in AAV gene therapy. The overall take-home message from our deliverable was, we highly recommend health authority engagement at each phase to get an early buy-in on your company's proposed strategy tailored to your product. It's really hard to have a platform approach. So having those conversations early on and through each step is highly recommended. So now we'll get deeper into the cell and the gene slide decks.
Okay, so I'll probably talk a little bit about gene therapy slide decks. So this deck, as already mentioned by previous already, is the deck is intended to share a typical QC testing panel together with the recommended controls and standard for in vivo gene therapy product. So based on the industry experience and our current understanding of regulatory guidance. So for a typical in vivo-based viral vector-based gene therapy product, a QC testing panel would include a biosafety testing, need to be tailored to its manufacturing production system, and then the measurement of the vector strength and potency, also the identification of packaged genome structures, as well as the capsid protein, as well as the evaluation of process and product-related impurity in order to ensure the product quality, safety, potency, and purity. So typically for a compendium-based or semi-compendium-based method, the commercial available standard come from ATCCs or other reputable vendors has been used in the assay. In contrast, the product-specific method typically would require a very well-characterized product-specific reference and standard materials. So, for example, can you go back to the previous slides? Yeah, for example, if HEC293 cells with transfection system was used to manufacture AAV product, then based on guidance document, then mycoplasma need to be tested in the harvest bulk to ensure the biosafety. In contrast, if SF9 and baculovirus system is used to manufacture AAV product, then per guidance document, because you're introducing insect cells, then not only mycoplasmid, but also spiravan plasmid also need to be tested in the harvest bulk to ensure the biosafety. So since AAV product tend to be able to store quite long-term at low temperature, typically a culture-based compendium method with appropriate positive controls would work. However, people start to explore a PCR-based rapid method even for a gene therapy product. In those cases, is as long as those newer, more rapid method has go through a formal comparability study in order to show the assay sensitivity and the string coverage are equivalent to the compendium method, then those methods will be allowed to use for the QC testing panel. So next slide, please. So for another example is people working with AAV knows the genome titer is a critical assay and be required to be fully qualified even for IND submission, is because clinical dosing will be based on your VG titers. So for this particular assay, very accurate and precise method will be required to measure the genome titer. So currently, two major platforms has been used to develop VG-based titer assay. One is a real-time PCR-based platform. For this platform, a very well-characterized product-specific standard or a very well-prepared quantified DNA-based standard, such as plasmid DNAs or G-block, would be required in order to quantify the genome titer. In contrast, the newer technology such as digital PCR will not require a separate standard for the quantification purpose. So that's also the reason digital PCR has become more and more popular in the gene therapy field for quantification. So gene therapy is a relatively new modality with several different manufacturing platforms. And then the lack of well-characterized universal controls and standard has been one of the major challenges to standardize QC testing panels for gene therapy product. So therefore, we hope that this deck can serve as a good starting point to help gene therapy developers to start designing its own QC testing panel. Thank you.
Excellent. And we'll give some examples from the CAR-T, Parul, if you want to.
Thank you, Kathleen. So going on from Jan, the CAR-T early and late phase, whether it's autologous or allogeneic, requires a profiling of certain attributes of the cell therapy that include genome stability, purity, identity. These are also safety profiles. And we also profile the quantity, that is the number of cells, the dose, and the potency. However, potency is not required during the early phase, more in the mid and late phase. Taking an example, you can see that most of, whether it's cell counts or dose, do not require controls usually, because whether it's strip and boost cell counting or VICEL cell counting or NC200 cell counting, that is an established process. Going to the next slide. Okay. So taking the example of genome stability. Genome stability is important because among the considerations for profiling these cells, we have to ensure that the manufacturing process or the cell culture or purification strategy for these cells did not affect the geneticity of the cell therapy. There are multiple ways where we can measure this. Here we are showing 3 of the methods. Fluorescent in situ hybridization maps or stains specific regions of the chromosome, and with gene-specific probes or target-specific probes. Again, we only need staining controls for these, as most of the staining probes are IVD devices. Then we have karyotype, where the entire 23 chromosome is profiled of the cell, and this gives more overview of the cell health and any large genetic changes at the macro level, that is changes in chromosome number or translocations or shape or structure of the chromosome itself. Should such a defect be identified, you can go and target using ddPCR or FISH these changes. So if there's a translocation, you can probe that region and see what percentage of your cells are affected to ensure the safety of your cell therapeutic. So as you can see from the karyotype, we do not have assay controls or alternative controls, whereas for the on-off target, as Jan was saying, we don't need controls, but if you have edited your cells, then you do need a positive, that is an edited cell versus a negative unedited cell as a control. And these can come from your pilot batch or engineering batch, or it could be a cell line, depending on what is being targeted by the ddPCR. And if there's a cell line available, again, in ATCC or a commercial source, that can provide such a translocation. Thank you.
Okay,
All right.
move on to the bridging and comparability.
Yep. So one thing that's constant is change, right? And I think with the pull 3 to 5 years, a lot of the different programs, you're approaching to that phase where things are changing. Either your process or your analytical is changing. So here we decide to review 3 common scenarios and offer additional guidance on how do you approach comparability or bridging. So in this scenario 1, you're faced with a situation where your analytical method has changed. First, you need to understand how does that change impact your historical data, and potentially how would it affect your specification of your product release. So a well-designed phase-appropriate bridging study is needed to test samples in both the pre and the post-change analytical readout. One common challenge that comes up during this design phase is the limited sample availability. I'm sure some of you have come about this situation, and it's always the more is the best. Depending on the outcome on the analytical bridging study, you may or may not need to change your specification. So just keep that in mind. And then in the next slide, we'll look at scenario 2. Here you're faced with a situation where your manufacturing process has changed or been updated. The guidance requires comparability studies to demonstrate that your post-change product is comparable to your pre-change. And just as important, you need to show that there's no adverse impact on your quality or safety of your product. So you'll need a comparability study design with statistical analysis and a protocol to evaluate how your post-change product compares to your pre-change. Of note, you need to know prior to executing the comparability studies that a post-change matrix qualification is required in order to verify that the analytical method performance is not compromised. Overall, the outcome of that study should be evaluated not just by meeting specification, but also against the historical trend of your particular product. And in our last scenario, next slide, yep. You're faced with both a process and an analytical method change. So this situation can happen by design or by unexpected events. One hypothetical example is maybe you're experiencing a discontinuation of an ELISA kit needed for an analytical release assay. Right about the same time, you're rolling out an improved process scale-up that was needed to meet the patient demands of your product. So in this situation, you must demonstrate both product comparability and analytical bridging to meet the regulatory requirements. In this case, you have several phase-appropriate options to tackle a combined study. You must determine which one works best for your company and for your product. We highly recommend, again, this is going to be a constant theme. We highly recommend meeting with health authorities during each phase of your product and discussing the study design in order to gain feedback. You never want to have surprises at the end, waiting till the end to file and then getting questions that will halt maybe your study. And then the next slide. So here we're just providing additional guidance that talks on comparability and bridging study dealing with cell and gene therapy products. The hyperlinks again are there for your review as a snapshot of the current guidance to date. Please keep that in mind again when reviewing the slide deck in the future. And I think that's our last slide, Kathleen.
I think that's it. So thanks very much, everybody. So I think this was a great starting point, and showcasing the deliverable was a good way to start the discussion. But I think the team agreed there's much bigger challenges in this area. So for the remainder of this webinar, we will look to have a panel discussion to address some of the sort of current and future challenges. We do have a number of questions already provided to the panelists, but we do encourage audience members as well to add some questions into the chat as well, and get involved in the discussion and give some feedback. So before we do, sorry, Parul, we do have one for Parul maybe. Parul,
Yes.
can you clarify the staining controls for fish? What should the controls be?
You should have like a positive control, like a housekeeping gene to show that your staining was not affected, and then a negative control where you have some bacterial gene or some other gene where it's not supposed to stain, because fish it's still a hybridization technique and it can be a little bit messy. So to ensure that the reagents you use and all your staining was carried out optimally, you have a positive and negative control. Does that help answer your question, Robert?
Thumbs up.
Thanks.
Thanks, Robert. And then we have a question for Shewit. For the purpose of comparability, do you have any insight for the characterization methods, tiering, and impurity methods?
Yeah, so that is going to be a company-dependent approach. Every product is specific and unique. It's going to be a panel, and I'm going to also open up to the rest of the panel if anyone wants to chime in here. I don't think there's one strategy that fits all. As long as you can have a strong explanation as to why you're proposing that strategy, where you can understand your product. It's always hard when you have to find what are your current quality attributes, particularly for your product, and can you compare your pre and post change? I will open it to the team, to the panel as well.
Brian, you're on mute, sorry.
Thank you. Yeah, I thought it was an excellent answer. I just would like to add to that, setting up the statistical evaluation kind of pre those comparability testing and really kind of establishing what some of those limits and what those statistical evaluators are going to be before we jump into comparability is always quite helpful.
So I did the report. I did the interim report.
Right.
So,
before we go into the panel discussion, we do have another poll for you guys, but we're interested to hear what you think are the main challenges when it comes to analytical control. So is it limited material? Is it identifying the correct control? qualification of bridging, we hear a lot, is a challenge, possibly limited stability of controls, or other. And if it's other, please drop into chat as well to just clarify what that is. This is a ranking question, so you should be able to shift your priorities up and down the screen. So if you rank what you think the highest challenge is versus the one maybe not so critical, and we'll look at that in a minute as well. That'd be great. Let me just see if anything's coming up yet. We'll go into the chat. We'll give people maybe a minute or two to join. So maybe I can ask to the panel what they think in terms of what the challenges are and the biggest challenges for securing analytical controls. Brian, if I put that to you first, have you any thoughts on?
Yeah, happy to. Yeah, thank you, Kathleen. You know, I looking at some of the early poll numbers. I'll take this as sweet validation. Some of the challenges with analytical controls, we could talk days about this, but I think ultimately limited material is the real driver. And one of the strange things, it's almost like on a frozen pond. It really just kind of it informs all of the challenges moving forward. After that, it's just being limited with the material that we have. And so when we kind of build onto that and really think about, how can we look forward and really kind of plan for some of that limited availability. Here at Bridge, we have an incredible transfer team, and their real job is to be Nostradamus, if you will, and to really look forward 18, 24, sometimes 36 months when we're thinking about where are we going to source our controls from, where are we going to get those from, how are we going to utilize those, but then at the same time thinking through stability of those controls and how we're using those and how long they're going to last. And I think really another challenge that kind of feeds into that is this idea of bridging is when we need to move on to that next reference control, however that may be, and again planning for that, ensuring that are we gonna pull a little material from our initial demo run, and then is that going to last us through setting up our analytical establishments and characterizing our material, and then moving through our various manufacturing processes and campaigns, are we going to be able to carry that control all the way through, or will we need to identify at what point do we need to then change material, supplement with a different material, and then including those bridging experiments and comparability experiments in an appropriate way to ensure that we're avoiding things like result drift, and we know that at each reference control that we replace is going to be comparable with the one before, so we can continue on with that. So again, the real mitigation I think is having a very strong transfer team. The team here at Bridge, we're staffed both with MS&T and AS&T scientists, and it's really nice. It provides cross-functionality, a lot of cross-pollination against groups, where we're always plugged into our processing sciences team as well as our analytical development team. So we're really kind of aware of needs, and again, that kind of forward-looking maneuver of knowing what we will need when, and then when we need to maybe make that change. Are we isolating enough of our original standard? We certainly don't want to exhaust all of our material, and then we get a phone call and all of our material is gone. We need to make sure that if we are anticipating bridging comparability, if we see a processing change or maybe even an analytical method change, those experiments that we have to run are going to be significantly larger than if everything stays the same. So again, having the right team that can really build onto that, look forward, and really plan appropriately goes a long way to mitigating some of these main challenges, again, of availability, and then ultimately bridging when we're ready to move on to the reference control number 2.
Thanks, Brian. It looks like the team agrees. So yeah, limited materials definitely coming up top. Then identifying correct control, qualifying bridgings number 3, and limited stability of controls. I don't know, both 1 and 4. Thanks, Ramashkin. There's a few people selected other, so I don't know if there's other challenges as well. Do you feel we haven't covered? Do you maybe want to drop into the chat? I can open it up to other members of the panel. Any other thoughts on challenges that Brian hasn't mentioned, maybe? Or do you think he's covered it all?
I think Brian did a great job. Very comprehensive answer. I think he touched on everything we were all thinking.
And guys, as well, just say if you think we've missed anything, just drop that into the chat as well, and we can pick that up. I've got one question coming in from Tony. What if assay control has stability trend over time? Anyone able to take that question?
That's a great question, and one of the advantages in my experience has been to place our material that we're going to be that we're using for reference control on stability, to be able to monitor that over time. It can be very difficult to say. I mean, it would depend on that trend and what we're seeing, and then ultimately what impact, what metric we're using that control for, what type, whether it's a ddPCR or an ELISA, what we're using that for. I would probably say, to maybe set up a formal stability study, try to establish kind of what those windows are, and then just kind of set up that stability window around that. We like to hope that we've got tons and tons of stability. That's certainly not always the case, especially with gene therapy products. But I think the key there would be able to evaluate that stability, determine exactly kind of what limitations we have because of the stability of the material, and then just set hard and fast controls, retest dates, or just replacing that material accordingly.
Thanks, Brian. I don't know if anyone else has any thoughts there.
Yeah, I would just say if you have to limit your control to a short time frame because of shelf life stability, it puts the planning even more at the center in order to, get batches more quickly, get bridging planned, because those studies take time to execute. So if you do have a limited shelf life, you really need to plan very, very well ahead, because sometimes those studies can take 2 or 3 months to complete. If you only have a 6-month shelf life, you constantly need to be identifying material and redoing qualification. So the shorter your stability, just the higher burden you have on those activities.
Thanks, Andrea. A few more questions come in. We'll put 2 more questions to the panel, and maybe then we'll go back to the panel questions. So any experience with functional cell-based assay control where relative potency is determined by parallel line analysis? Andrea's nodding there. I don't know. Who wants to jump in?
So typically we don't do parallel line analysis. Usually if we're doing a functional cell-based control, we usually do 4-parameter fit with the ratio of the EC50s, but we do certainly have cell-based relative potency assays where we use the reference standard as a calibrator. So I don't know if you have a specific question that you would like to ask around that use.
Feel free to come off mic and expand on your question if you like, Kirill.
Hello, this is Kirill. Just can you say again how your controls work? So you have standard curve, and you don't, but your sample does not have standard curve. Is it?
Yeah. So instead of doing a parallel line analysis, we use the 4-parameter fit. So just a this sigmoidal curve instead of just the linear portion to do kind of the EC50 or slope analysis.
I thought parallel analysis required 2 sigmoid curves.
Yeah, two sigmoidal curves, but instead of the parallel line analysis and the EC50 analysis are a little different, although very similar. So we might be talking about the same thing.
Okay, thanks.
And we have another question from Seema. Should the options be A, qualification of assay control, raw material, and B, bridging strategy instead of qualification of bridging?
That's a fantastic question. And what, in my experience, what I found is that's going to be driven firm to firm, and your timelines are going to drive some of that. You'll need to risk assess some of how you're doing that, and really kind of build in some of that risk, and really kind of understand what you would accept when it comes to risk. But I think that, if you can do some of this concurrently, when we talk about, qualification of the reference material, and then being able to then use that material ultimately for your bridging is probably best case. But in my experience, this type of question is usually driven by the firm, kind of internal procedures, which I guess ultimately is going to be timeline driven on, again, how quickly we can get some of this achieved. Whereas we're looking at some of the methods and how, again, these needs that we need, as Andrea had mentioned, sometimes, some of these, tests and types of analyses can take some time. So we really need to build some of that in when we're kind of risking out the potential of what needs to be done when. Can we do it in parallel, or do we need to do things in series?
Great. All right, we'll go back to some of the questions we have, and then we can drop back into the audience questions. So what are the key safety parameters accepted by industry and health authorities for cell and gene therapy products? Jan, I don't know, do you want to maybe tackle that first?
Sure. I probably more comment on the gene therapy side, and then I can leave my colleagues to comment on the cell therapy side. So for gene therapy, particularly currently most popular class of product is AAV-based gene therapy. For those, my view is very similar kind of guidance document in the, guarding the safe, at least particular biosafety side, has been used for many years for recombinant protein. The principle there is basically you're either looking for your production system. If you're using cells, plasmid, virus, helper virus, or so any particular raw material to establish your safety parameter, depend on the starting material, and those guidance is very similar as the guidance to guard recombinant protein. You're looking for your two-tier cell banking strategy, have mass cell bank, working cell bank, and cell bank has to be extensive characterized. And then depend on your plasmid and virus, then you have the similar characterization. Also is the bulk harvest control cells. Those I think is very similar as a recombinant protein side of the business. The only thing different for the gene therapy per se is because gene therapy does use a different kind of type of cell line. As I mentioned in the earlier presentation, is you need to consider if you use inside cell line, then your safety parameter testing has to be tailored to inside cells. They have their unique virus, particular endogenous virus, then you need to add additional safety testing into your QC release panel. And also because if you're dealing with a viral vector, then you also need to add a replication competent viral testing. So this I think is something unique. And then there is a standard methodology across industry that you need to add that one into your testing panel. In terms of product specific parameter guard safety, for AAV vector right now, I think regulatory authorization start to looking for total particle. So typically people started adding upper limits of a total particle allowed in your final dosing to increase the safety margin there. And also is for the whole cell protein, whole cell DNA, those things has similar to recombinant protein. But I think for the residual whole cell DNA right now, FDA start to lose up a little bit on the limits. You can do case-by-case justification if your DNA is greater than 10 nanogram per dose. But you do need a certain data package to make sure the sizing distributions and whether you have any particular interesting sequence in your final product. So those I think is, but FDA is not going to using 10 nanogram per dose to as a hard standard. Also is for gene therapy right now, the empty was full particle is become another parameter kind of guard to product specific safety, even though there's no clear guidance document to say how much empty particle is allowed, but you do need to add into your release panel, have a good understanding what is your product, how much empty is in your product to at least to keep it consistent. So I will stop here, ask Parul to comment on the cell therapy side.
Thank you, Jan. So on the cell therapy side, usually all the starting cell materials are either cultured or purified, and any media or raw material that has a biologic origin, that is like FBS or serum or growth factors, need the source animal virus testing to be completed. So if you are taking, say, FBS, then you have to check out the bovine virus. If it's got a mouse antibody, there are a panel of mouse viruses that need to be profiled in your cell therapeutic. If it is porcine, same applies, like you have to rule out porcine viruses. So the take home is use well-defined media or profile the raw materials. And sometimes that gets really expensive. So this is something that you take into consideration during the manufacturing process optimization. Another consideration for all cell therapy is also, from the safety point of view, sterility. That cannot be done like for biologics or small molecules. You cannot filter or sterilize them in the normal way. Here's what I found. Sorry, that's my watch talking. But you have to ensure that your environment for manufacturing is does not add any endotoxin or bacterial or fungal contamination. So the clean rooms play a critical role in cell therapy, um, which is very stringent, like how you gown and how you monitor the person who's doing the manufacturing in the BSCs. And that's very typical of cell therapy, unlike for biologics or small molecules.
Thanks, Parul. We've got a couple more questions in the chat. So would it possible to submit limited stability data to support filing, example BLA, with commitment to provide other data afterwards? I don't know if anyone has experience in that and can talk to that question.
So I will say, yes, you can, but you're limited to whatever you filed. So until you show the additional data to support extension, you are limited to what you filed. So that's also something that's possible.
Yeah, I concur that one, because particularly for BLE, you do need to define the shelf life based on the data, based on extrapolation, whether you can claim. Because from commercial side, if you have very short shelf life because you have a limited data, it's very hard to manage the supply side. So there is a criticality there.
I just also want to point out here, we're talking about assay controls here, and even if you're using a product as an assay control, they don't necessarily need to have the same shelf life. You might find that you want a tighter control on your assay control, especially if you're using it as a calibrator. So it may have a shorter shelf life than your product, or a longer one, depending on what the data and the assay use is. So just I want to make sure we're thinking of the assay control as a separate entity than the product itself.
That is a good point. So basically for assay control, typically you have a lot more flexibility, and also your container closure is different from your final product. And typically you're going to store them at -65. So with that in mind, I think one practice you can consider is don't even assign expiration date, rather assign retest date on your COTs. That way it's give you even bigger flexibility to extend the expiration, or extend the usage of your precious material as long as you can afford.
Great. Thanks, guys. I see there's some good conversations as well going on in the chat back and forth, so that's great to see as well. We'll come back to maybe a few more of the questions that's been put initially to the panelists, and then we can come back and pick up on the chat as well. So in terms of new technologies and upcoming technologies that we need to consider, what are these for the analytical controls? So Andrea, maybe, do you want to take that?
Yeah, so I'll just touch on something Jan already talked about, which is the move from qPCR to digital PCR or droplet digital PCR for gene therapy. This does allow you to get rid of your reference standard, so you get an absolute quantitation, which is great as far as bridging, and you really get away from that needing to bridge standards and worrying about assay drift. However, in those assays, you are still going to want to have a product-specific positive control to make sure that the assay is giving you expected results as you go. The other gene therapy control that I'll touch on, that as technologies kind of evolve, I think the full control and the empty control for empty full methods are becoming more critical, and those can be hard to manufacture. There are some commercial vectors available, but as you're trying to match it as close as you can to your vector, being able to produce full and empty kind of pure controls for assay validation is becoming a challenge as well.
Anyone else anything to add there? Or audience members, any thoughts? We want to drop it into the chat as well. A few more questions coming in. Can you simply test for murine virus using MAP or MAP?
I can take a step on that question. So I think, I guess the question, I mean, so this is come from CHO-based cell line. People has been using MAP to test marine virus, but because CHO cell line has not been heavily used in the gene therapy field, so I don't think that necessarily direct apply to gene therapy product biosafety testing. And in addition, there is a 3R, heavily effort on 3R. I think more and more based on the recent guidance document changes and then the 3R restriction, I think more and more people will stay away from in vivo animal-based testing and move towards to sequencings or NGS-based technology.
I agree with Jan, because also the sample requirements or materials are too high for doing individual viral testing. So the next generation sequencing will be the way to go, with a large part of your cell therapeutic gets utilized in the adventitious agent viral testing.
And we have a question from Karen. Hello, Karen. Wondering, what is the justification for less than 1,000, or less than 100 CFU for bioburden in CGT when sterility is critical at drug product? Wouldn't you expect a lower bioburden prior to sterile filtration? Don't know if anyone can take that.
Is it just less than 100, or was it 10 to 100 CFUs?
Yes. I was
trying to remember what was on your table. I'm sorry. I thought it was less than 100 CFU per mil, probably, right?
We can go back and confirm that maybe, and we can come back to you, Karen.
Yeah, just curious.
Maybe
trying to keep everything as low as possible before filtration, right?
Sure. Okay. We'll confirm that.
At the other end, for cell therapies, we have less than 0.25, like endotoxin levels are usually used to measure sterility. The bioburden is more to show that your cell therapeutic can support the organism being tested. So the CFUs are like you inject the organism and show that it will come up positive at a certain level.
Right, for a standard or a control, correct? Less than 100?
Yes.
Yeah, agreed.
Yep.
Agreed.
Yeah,
just wondering if that's your limit, your spec limit on maybe DS.
The golden standard is negative bacteria, negative sterility for in AST, like, for aerobic or anaerobic or fungal organisms in your cell therapeutic.
Okay.
Okay, another question to put to the panel. So what is your typical approach to generating analytical controls? So again, Andrea, maybe, or do you want to take that
one? The key with an analytical control, especially when it's a product-specific control, is that you want it to be representative of your process. So this could be taking a full-scale batch out of manufacturing or an engineering batch. With the small batch sizes of cell and gene therapy, typically you may require an entire batch. This goes back to planning. As Brian was saying, if you need to be using that full-scale manufacturing facility, you really need to plan ahead and make sure you can get batches made. But really, the key is to have it be representative of your final process and of the quality attributes that you expect from your product.
Anyone else? Any thoughts there?
I guess I would just add, thinking about the metric that we're using to interrogate our analyte sometimes too, it seems gene therapy can change with warp speed. You know, I mean, and what we're looking for. I guess an example that I can think of is residual plasmids. It wasn't that long ago where it was just residual plasmids, and now it's encapsulated residual plasmids versus total residual plasmids. And now there's a lot of interesting discussion about capsid-associated, plasmids. You know, plasmids are genome fragments that are somehow associated with the capsid and still might show some potency, might still have some relative potency. So again, I think that one of the things is also trying to stay nimble and ensuring that we're using our controls in the appropriate way and looking at the right metric, determining on what we're looking for in the assay itself.
Yeah, to add Brian's point of view, I also want to share a little bit is for product specific controls and the standard. I think there's a terminology has been sometimes used differently or similar in the field. I think from technical standpoint, it's be beneficial to really understand what you're using this product specific reference material for. Because for certain assay, let's say for potency assay, if you're using relative potency, that in my mind is the most important because it's a standard because your value is based on this material. In contrast to other cases, we just want to control the assay performance within the trend to other part of system suitability. So in my mind, even though they all product specific reference material, but the requirement is slightly different. So I think if you can set up your SOP internally to differentiate those, you may be able to use your most precious, well-characterized material to the best you could, and then downgrade the other just simply to control the assay performance, but the number is not relied on this particular material. So I will stop here. So.
Two questions just in the chat. So for safety, example, live contaminants, what is the most recent strategies to replace in vivo with molecular assays?
So I will try to, so probably I need a little bit clarify this question is, in the typical biosafety testing, the only in vivo assay people still are using probably will be strict to mass cell banks or mass viral bank. Because based on current guidance document for anything like working cell banks, working viral banks, derivative from well-testing masked material will now require go to in vivo testing already. So I'm not sure the context of this question is related to mass cell banks or is related to derivative material. So I probably need a little bit more clarity on this question.
Jennifer wants to quickly comment.
Yeah, it's like I would like to know the trends of the strategy in analytical section. Seems like during the novel guideline, we're trying to move from in vivo, all kinds of in vivo testing to like maybe molecular assay or even NGS. So I just want to know like your opinion regarding like how we can do that. Thank you.
Oh. I think I used to work for Charles River. I believe Charles River recently published a very interesting paper to compare in vivo viral testing with NGS. I have not read that paper, but if you have time, have interest, maybe you want to take a look to see whether give you a little bit more in-depth information. Just a suggestion.
Thank you.
We've got 1 minute left. I appreciate we're probably not going to get to this last question, and any questions we've missed, we will go through and respond offline. So thanks for the questions, guys. Just very quickly, just to flag this, in addition to this deep dive team, there's a number of other deep dive teams within the forum. You'll see some of the topics that are covered here and the modalities they span. If you're interested in any of these topics, please reach out to me and I can put you in touch with the relevant people. Also, just to say, this is one in a sort of a series of webinars coming up. Our next one is next week, and it'll be focusing in on viral clearance in cell and gene therapy, and we'll be showcasing a recent publication from another deep dive team on that topic. And I think hopefully one of my colleagues is going to drop that registration for that webinar into the chat. Final words, I don't know, before we go, any of the panelists have anything else they'd like to say?
The field is evolving. We just gotta, yeah. Thank you.
use the slide deck and give us the feedback.
I think
Yes.
it might be updated. A
Agreed.
live document might get updated again. Okay, thanks everyone. Thanks for your participation. Please do download the deliverable if you haven't of yet, and we'd love to hear your feedback. Thanks to all the speakers too. Appreciate all your support.
Thank you.
Thanks all.
Thanks, guys.
Thank you.
Thank
Yeah,
you.
take care.
Talk soon.
Bye-bye.
Bye-bye.
Thank you.
You.